Hey guys, so this week we are discussing oncogenic viruses, or sometimes they're more referred to as tumor viruses. Um, what is often kind of relegated to more of a side portion, if that's the right word, of virology. It's at least not as common, but it just sort of depends on the field. But actually, oncogenic viruses are super, super important and really common. We're going to see that. A shorter lecture this week, I wanted to just touch on some of the big stuff with oncogenic viruses. So, for a set of viruses that we don't spend too much time talking about, it's actually a huge deal. So there are 8 big families, and we'll talk a little bit more about what that really means. But you can sort of lump 8 different viruses or their families of viruses into 10 to 12% of all global cancer. All cancer. Like, that's not just of cancers caused by an infectious disease or something. No, like just all cancer. It's just crazy. That's so high. Um, it kind of depends, but in general, the poorer areas of the world and more marginalized groups have increased burden. So you can take that economically. So you can see on this chart, which is an older chart, but it still basically holds true. The poorer areas of the world tend to have higher disease burdens. It depends on the disease. This is a summation of tumor viruses. But often what we're seeing here is HPV is a big one. There's other viruses too, but what we're seeing too here, especially with Africa, is the presence of HIV suppresses the host immune system and allows tumor viruses to do a better job at what they do, which is make tumors. And so that's where you're also seeing some of that disease burden. We'll talk a little bit about how HIV influences tumor viruses and tumor virus disease. Hence often associated with immunosuppression. But we do see it in other marginalized groups. So men who have sex with men. Australia has an issue with indigenous peoples. Um, I guess Mongolians. That's like super red. That's Mongolia, right? That's kind of crazy. Why is it so red there? That's a good question. So anyway, but it is a huge problem, and let's talk about it a little bit. Now, this next chart, it's from that same article. So if you want to head to the article, it's a really good summary of uh, basically human tumor viruses. There are non-human tumor viruses, but we're going to talk humans today. I like this chart a lot because it gives the years on that. There's just a ton of info. This is only, there's actually a few more lines to this chart. If you actually go to the website, I'm not going to put it all in here. But it's a nice summation of what's happening. It's got the year was discovered, the types of cancers that are associated, which is really, really helpful, and the disease burden. So some of these actually are pretty low. Now, keep in mind, this is per year. So HTLV1, which I'm going to talk about in a little bit, only 3,000 per year, but that's 3,000 people whose lives are ruined every year because they get a T lymphotropic cancer. And it's usually really aggressive, so it's really dangerous. But you can see global prevalence. HPV is huge, and Epstein-Barr is huge. Um, those are so Epstein-Barr would be a herpes subvirus. What else? Just there's a lot of cool info on here. Routes of transmission. Where is it highest prevalence? Um, do we have much knowledge about it? That's a really good question. What's actually causing the virus to perform oncogenesis? Because we really have two things that we're dealing with here, right? We have a viral infection. Right? That's what this class is, right? Virology. But you also have oncogenesis, right? And so what advantage is the virus gaining from causing oncogenesis? Do we understand how it's actually causing cancer? That's a good question. A lot of times we don't, but sometimes we do, and it just kind of depends. I do like this too because the years that they were discovered are really interesting. The history of tumor viruses is really interesting. One of the review articles that I have for you guys is about the history of tumor viruses. I'm going to go through a ton of the history. That paper is really good. I have the reference right here, and it was given to you. I grabbed these 2 timelines. It's one long timeline from the paper because it's really, really good. And I just wanted to emphasize the craziness of scientists. So I've been in the world of HIV for a long time, and it's the same way where if you try and break the paradigm you are going to get blasted from all these people who've been doing it for their whole lives, which I kind of get it, like, hey, this young buck's coming in here and he's telling us this thing might be different. Well, you know what? That's science, and you're supposed to question either way, right? And so if somebody has compelling data, then you need to take that compelling data in and use it to, change your own science. But that is a tall ask for a lot of people, and that's what happened to many, many researchers early on. So once we were able to, we knew what viruses were, but the idea that a virus could cause cancer was very, very up for debate early on, especially in the tens to 20s to 30s. It was a rough time if you were Rouse, basically. So we now have one of the most important viruses that causes cancer ever, Rouse sarcoma virus, was named after Rouse. And so you can see here, that's this one right here, the avian Rouse sarcoma virus, RSV, which is different from respiratory syncytial virus, which they both have the same name, which is super annoying. But he discovered that in 1911 and was not given the Nobel Prize until 1966 for that discovery, because they basically just were like, that's dumb. Yeah, you're full of it. And they just tried to discredit him all the time. I'm sorry, this looks weird. My bad. And so just keep that in mind. If someone has compelling data, you can check up on their data, but you got to take that in and incorporate it. Tons of great research was done, though. You can see there are way more things here from the late 60s through now, as opposed to back here, because we were just not doing the research. Now, part of that was that the model systems weren't great, or some of the techniques weren't super great, but actually a lot of it had to do with just dogma. People did not want to accept the idea that viruses could cause tumors. We didn't really understand cancer super well back then either, so there's just a lot of issues. It turns out cell cycles and the causing of cancer is really, really complex, and cancer is such a broad term for arguably thousands of different diseases, and so you can see why there was some pushback. But anyway, great paper. You should read it all the way through. It's really interesting. Now we can basically take our tumor viruses and put them into two categories: RNA tumor viruses and DNA tumor viruses, which you can basically put any virus into one of those two categories too. But as far as tumor viruses go, that's a really, really important distinction. So tumor viruses, RNA-based tumor viruses are retroviruses, with the exception of hepatitis C. Which, keep in mind, all that there's hepatitis C, there's hepatitis A, there's hepatitis B, there's hepatitis D. I think there's one more. Is it hepatitis E? I can't remember. Those are all very, very different viruses. They just happen to infect the liver, hence hepatitis, but they're all really different viruses. They are basically unrelated, though I think B and D are related. But all retroviruses, just like HIV is a retrovirus, where they take their RNA, convert it to DNA, and then embed that into the host cell. So the previous lecture that I did was talking about latent viruses, and that would be an example of a proviral latency, where they're going to take their genome and insert it into the host. Now, HIV is kind of the poster child for retroviruses and this kind of proviral latency in general. HIV, I mean, there's strains of HIV, doesn't typically cause cancer itself. It usually influences other viruses to cause cancer. Now, hypothetically, I've seen examples of HIV-induced cancer, but it's pretty rare because HIV is kind of random with how it actually attacks the cell and where it inserts its host genome. So the only time you're really going to get an HIV-caused cancer is when you're going to accidentally embed your HIV provirus into a tumor suppressor gene. And that's only going to be in one cell. So honestly, it's pretty rare that you see that kind of thing. But the actual influence that HIV has on the rest of the host can cause cancer. So most of the retroviruses, mostly RNA tumor viruses that you're going to see are retroviruses. And they tend to drive the cell cycle forward. And so what do I mean by that? Well, that means they're usually influencing cyclins or cyclin-dependent kinases or something that causes the cell cycle to go forward. So cells, there's a common analogy that cells are like a car, and cyclins and cyclin-dependent kinases are like the gas that's driving the car forward. And tumor suppressors like P53 or P21, there's a bunch of tumor suppressor genes, RB, those are like the brakes. And so if you're kind of in a balance between those two things. So you want your race car to go a certain speed, but if you start going too fast, you get a little crazy, right? So you need to put the brake on a little bit. And so cells are very much like that. And so you can break one of those, and then your cell is just going to go crazy and go wild, and you're going to crash, i.e. cause cancer. And so RNA viruses tend to push that gas down too hard. You can push the brake down, but you're still going to crash, right? DNA tumor viruses is the other side, and they tend to disable the brakes themselves. And so you've got the gas down at like 60 miles per hour or whatever, and you disable the brakes. Well, you're going to crash at some point, right? You have to stop at some point. Whereas RNA viruses might not influence those. They don't affect the brake, but you can stomp on the brake all you want, and if the gas is pushed as hard as you can, then it's not going to make a difference. And so that's what we're kind of organizing these two things. And this is a picture I took from the cell cycle. You've probably seen it before if you took genetics with me or biochemistry with me. But this is just a really good diagram of the cell cycle and the different cyclins and cyclin-dependent kinases. So we can see in the gray, I don't know what they're, curvy things here for cyclins and cyclin-dependent kinases. These are what drive the cell cycle forward, and if you push hard enough, you can get them through the checkpoints. But the brakes are all these crazy things here. So you got your P53, your P21, and they're checking, in general, there are a lot of these things right here, like BRCA and ATM and stuff, they're looking for mutations or things that show up, DNA damage, if your cell is damaged, if your checkpoints aren't proper. So like you're not checking your spindle fiber formation, or you're not making sure that your DNA has been properly assembled or doubled during the S phase, just things like that. And so that's how we separate our two tumor viruses. Now, I took this from that same article about the history, just to kind of show that there are tons of tumor viruses now that we know of. And what's really interesting is what kind of, so they're causing cancer, right? It's a virus infecting a cell, that's one thing, but they can cause a cancer. And so the actual viral proteins that are affecting the cell and causing the cell to become cancerous vary widely. I'm going to talk a little bit today about HPV and HTLV as just some examples of an RNA and a DNA virus. But like the E6 and E7 protein are really important. You can see HPV here. It's a DNA virus that's going to affect P53 right there. That makes perfect sense. You can see that a lot. P53 here for SV40, which is in monkeys. P53 for adenoviruses. P53 for HPV. There's other P proteins too. RB is the this guy right here that suppresses the cell cycle during this step. So if you break that, you're going to pass through there. And adenoviruses affect that, as well as polyomaviruses. And there's lots of different viral proteins. So there is some consistency between viruses and the proteins they affect. But there's a lot of differences too. And so, yeah, it's just really, really interesting. And so there's a ton of space in the literature too. We just don't know a lot of these things because, let's be honest, it's hard to study some of these things. Yeah. Of that, we're going to start, I'm going to give two examples today. I want to talk about HTLV, which is human T lymphotropic virus or T cell leukemia virus. The L gets swapped around a lot, so that's HTLV here. And I want to talk about TAX. So HTLV is a retrovirus. I apologize for the quality here. Let me shrink it a little bit, make the quality slightly easier to look at. So it is a retrovirus. It's one of the earlier retroviruses that were discovered. And so it's going to take its RNA. The only reason I really picked this one, it's super busy. I realize that. So just follow me, okay? So we've got our virus. It's going to get in here. It's got its single-stranded RNA, converts that using reverse transcriptase, which is what HIV uses, into single-stranded DNA. Right? So there's it's single-stranded DNA. It's negative sense is what it's trying to imply here. It should say ssDNA, just like ssRNA, but whatever. And then it goes to double-stranded DNA. And that's what actually gets inserted into the host. So that's what's in brown here. Why they pick brown, I don't know. And it's brown as being embedded. There's the long-term repeat. And you got a 5' 3' end, and it's embedded now in the host. And the host cell will have that inside of itself forever. The only way to get it out, to get rid of it now, is to kill the cell. So there's a lot going on in this chart. So when you start digging into HTLV and HTLV's life cycles, you encounter a lot of really interesting stuff. So if you want to fall down that rabbit hole like I did, go for it. But the moral of what I want to talk about here is TAX. So TAX is a protein. It's a viral protein that gets expressed and has a ton of different effects. Rex is another one, but TAX is the other one. And this article in particular is talking about the connection between HTLV1 and microRNA and its control over the microRNA pathways, and its ability to control risk, which is part of microRNA, and its control of risk and how it uses risk to actually affect the cell and cause tumor. But the key here is TAX, and so I want to talk about TAX a little bit. Man, the quality here. Some of these articles just didn't have the greatest quality as far as pixel count pictures, but I did like the pictures a lot because they talked about things that I want to talk about. And the main thing I want to talk about with TAX, so this is a viral protein that HTLV makes. And what it does is it primarily, so remember, this is an RNA virus, and RNAs they push that gas pedal down so hard that you're not able to suppress it. Now, it turns out that TAX actually does repress p53 function. So that is a good thing for the virus, a bad thing for your cells. And so remember when I said this is what RNA viruses and tumor viruses, why they're different? Well, it turns out there's a crossover between those two. So TAX affects p53. Oh, okay, well, that's different. But main thing here is that it's going to drive the cell cycle forward. So you can see here we're going from our G1 phase to our S phase. So it's able to influence these cyclins and cyclin-dependent kinases right here to actually pass. So like CDK2 and cyclin E. It's able to push us through that cell cycle checkpoint, which the more, with cancer, the more times that you are able to get one single cancerous cell to replicate itself, the more likely you are to gain more cancer-causing abilities, basically. So every time you get that cancer cell to replicate, it gains more oncogenic abilities. And so if you can sort of get it over that hump, that initial hump through these cell cycle checkpoints, then you're going to be able to engender more cancer attributes. And it's really, really interesting, especially some of the influence that these viruses can have on like angiogenesis or metastasis. So you have to think about it. It's really, really logical. And with HPV, it's going to be even more logical. But to have a cell become cancerous that's infected with the virus, well, all of these cells have this provirus stuck inside of them. So if I replicate the cell, I'm therefore replicating the virus. So it's a way to just sort of spread the virus around, but you're kind of taking advantage of the cell. So you're sort of using cancer as your vector as opposed to virions, though you do both. So let's move on to tumor viruses. So you can see, this is a nice little picture I got from somewhere that it talks about tumor viruses, and you can see the suppression that they're carrying out on the tumor suppressors. And so this is the key here. So these viral oncoproteins, which usually they have multiple functions. I mean, TAX is a single protein that has like a million functions. And so viral oncoproteins, in this case with DNA viruses, are usually affecting your tumor suppressors. And your tumor suppressors, remember, are those brakes. So if I break the brake, it doesn't matter how fast I'm going, eventually I'm going to crash. So a great example of that would be HPV. So human papillomaviruses, and then other DNA viruses too. The most common ones would be like your herpes viruses. So your Epstein-Barr, your genital herpes, your cold sores, HHV1. They usually have episomal latency as opposed to proviral latency. Okay, and so a lot of these viruses are latent. And so classically with human papillomavirus, so every wart that you've ever seen, like if you have a wart on your hand, or if you get genital warts, or you can get warts on like your face, you can get genital warts like on your lips, that's caused by a papillomavirus. And human papillomaviruses are the ones that affect humans. Duh. And there are papillomaviruses that affect all kinds of creatures. I saw some for like turtles and cats, and like basically every animal on the planet has papillomaviruses. but they cause warts. And so warts are a form of cancer. It's usually benign, right? Like you can have a wart and it's not a big issue, but it is caused by a virus. And I like this article a lot. All the pictures that I have here, the next two slides of pictures are from this article. It was a really, really good review article. There's some really good review ones for HPV because HPV we see a lot in the news, whatever that is, because of cancer, right? And the vaccine that we have against it. If females get HPV 16, so strain 16 or 18, then that covers, I think, about 60 some odd percent of all cervical cancers. So it gets on your cervix, it causes an infection, and it usually has to be inserted into there. And so there's usually like some damage. And so that's how papillomaviruses actually get in. They're non-enveloped. And so they don't have that nice squishy phospholipid bilayer envelope to actually enter a cell. And so it's usually from damage. So I had a papillomavirus on my foot. I had a plantar's wart, so it was still a wart. It was a papillomavirus, and I got it from I was in the bathroom. I was home from college, and I stepped on a toenail that my dad had left on the bathroom floor from cutting his toenails. Because I don't know, my dad and I shared a bathroom, and I was in college, so he didn't clean up very well, and it poked into my foot far enough, and apparently there was papillomavirus on that toenail. I know, it's super gross, and I had a horrible papillomavirus infection on the bottom of my foot. Anyway, but you need some kind of wound, which is actually very similar to other non-envelope viruses, especially for plants. That's the next that's the next thing we're going to be talking about in this class is plant viruses. They enter via wounds, and so you need some kind of vector. Either like a physical, like an inanimate vector, like a nail or a toenail, or it could be an animate one, like from like a creature, like an aphid. I don't know why I have like an aphid in my head. Anyway, so you have to get it in there, and you want to get those basal cells. If you're the virus, you want to get those basal cells infected because those are going to be your like precursor cells, so they're stem cells basically, and then they're going to be infected, and then you keep your episomal virus infecting these stem cells, so it's sort of living inside here, and then as those cells differentiate, so we start making our lesion as it keeps growing, so this is the actual wart part, okay, and then so you got your nice wart lesion here, and these cells at the top of the wart, that's where you're actually going to get the virus being made, and so because that makes sense, so you sort of use the stem cells as your growth medium, and then you make actual virions and get those to go infect new hosts and new host cells from these top cells here. And so that's one way that a non-enveloped virus is able to spread using an envelope in a way, because it's sort of like keeping an entire cell around itself instead of just like a viral envelope, which is really interesting. The idea of these becoming latent is really, really important, and so you can develop latency for papillomaviruses, so you don't really have a wart there, but the basal cells are still infected, and so the idea of how those can become reactivated is really interesting. Reactivation from latency is a whole field of study. Now, this is from that same paper, and I just wanted to address some of the ideas that are coming from it. So you can see we have RB here, so that's a tumor suppressor, and we have P53 down here, that's also a tumor suppressor. So we know that typically, unlike RNA viruses, which drive cyclins and cyclin kinases, usually your DNA viruses are just going to disable the tumor suppressors, and so we can see that here for HPV. Now, you can generally classify HPV into either high risk or low risk, the risk being cancer. And so the high risk ones, you can see they do a little bit more, and they tend to go after P53. And the key to high risk HPV infection, in case you were wondering, some of you want to be doctors, is that the high risk ones will express E7 and E6 proteins. Which, if you really recall, over here there's E6 and E7, and those are the ones that are going to be the powerful suppressors of the tumor suppressors, if that makes sense. Notice here as well, they're also able to upregulate cyclin, Okay, and to drive the cell cycle. So, once again, when I said, "Hey, this is the difference between RNA and DNA tumor viruses," yeah, that's it's. I don't know where that distinction came from. I'm only really telling it to you to just sort of keep it organized in your brains, but obviously all the viruses do all of the things. So, what you gonna do? But you can see down here. Um, so, hang on. I have the little cheat here. I always forget what CIN stands for. So it's cervical intraepithelial neoplasia. I wasn't going to show you guys that, but so there's different categories. So if you get diagnosed with cervical cancer, I mean, you have to be female to have that happen. As a side note, HPV causes a ton of cancers in males and females. That's why the actual HPV vaccine should be given to every male and every female all the time. The only reason we don't give it to like little kids, like my little kids, you're supposed to be like 12-ish, 12 or 13 is usually when females get it, is because it makes adults uncomfortable to give their young children a what's arguably an STD. HPV is often sexually transmitted, so it feels weird to give your 5-year-old an STD vaccine. But honestly, I don't know. It's just protecting them from the future. They could engage in sexual activity later on in life with somebody who has this disease, and so you're just protecting them. People are just really weird about sex in this country. Is basically what it's trying to say. And so, if you get diagnosed with cervical cancer, if you're a female, some of the other cancers that cause, I mean, there's anal cancers, penile cancers, oral cancers, all caused by HPV. Um, and so you can see here, the really bad ones have E6 and E7, right? That's your high risk. And so down here is a really cool chart from that paper. You've got your basic infection here, which it looks like these same authors. And then you have your infection that's making it's cancer and it's making infected cells that are making virus. And the arrows over here are the different proteins that are expressed. And so E6 and E7 is the key one here. And so you can see as the bigger the arrow, the more the expression. And so you can see as we travel on, what is it? Cervical intraepithelial neoplasia. Basically, as your cancer gets worse, the expression of all of these proteins goes up, especially E6 and E7. And so that's when you're getting these really, really dangerous cancers that are going to then metastasize and spread. If you want to dig deeper into actual cervical cancer, it's out in an entire world. One positive thing that comes out of the vaccines, though, is that you can have cervical cancer, and you'll see these kinds of terms like, oh, we took a bunch of women who have CIN level 2, and we gave them the vaccine, and it dropped them from CIN 2 to CIN 1, or from CIN 1 to a silent infection. And so you can actually have repression of the cancer using a virus vaccine. So that's kind of awesome. So it's an anti-cancer vaccine then, or like an oncogenic vaccine. The reason that happens is because we're taking advantage of the fact that these cells express HPV antigens. So vaccines make antibodies against antigens, right? Or T cells that fight the antigens of a virus. And so if you can these cells express viral antigens, and therefore the immune system can attack them. So that's different from other cancers, and so that's just the advantage that we get out of that. Just a little bit about HPV. So just in the U.S., this is a 2012 paper. A lot of the stuff is from 2012 and 2014, because that was around when we started doing more with the vaccine, at least in the U.S. But you can see these are all, everything in red here are vaccines, and the number of annual cases of cancers, the number of cancers caused by HPV. And so you can see, like, if we would get everybody to take the HPV vaccine, we could completely eliminate cervical cancer. You could eliminate almost all of anal cancer, at least over half of vaginal and vulvar cancer, vulvar. I don't say vulva very often. Penis cancers, oropharyngeals, and so this anal cancer one is actually huge. It's a huge problem in men who have sex with men. And so that's a big deal. So that's actually a second group, an underrepresented group. If we go back to talking about the disease burden and marginalized group, well, you can't get much more marginalized than homosexual males or men who have sex with men. And so that's why there's a huge amount of anal cancers from HPVs. Now, the success story really is in Australia, where they required vaccinations in females, and then I think they required it in males as well. And so this is between these two lines is when they initiated the vaccines for on this side is females and this side is males. And I just wanted to show you the disease burden for indigenous versus non-indigenous peoples. So that's like the Aboriginal peoples in back Australia, and you can just see the number of genital warts, which connects to the number of cervical cancer cases as well. It drops drastically in younger people, which is awesome. That's what we want to see. So you start vaccinating the young people, it turns out it makes the infectious disease go away, and then people aren't dying from cancer, and we don't have to pay for people that are dying from cancer, right? If you want to look at it from a suffering standpoint, awesome. That's what I encourage. If you need to look at it from a purely economic standpoint, all these infectious diseases that viruses cause are huge economic burdens. And so by eliminating those, we can save a ton of money, which would be great. Also, people don't die, which is also a good thing. I just wanted to talk a little bit, we're actually almost done, just the different kinds of viruses. I just like this one because it's got all the proteins that each of viruses. So this is 1, 2, 3, 4, 5, 6. Between 6 and 8 viruses is what we usually talk about as being the ones that cause these viral cancers. Like this one's excluding this last one, Merkel cell carcinoma, because that's a relatively new virus. But this is just kind of a cool chart where it talks about like over here is like anti-apoptosis, so the cells aren't dying. Angiogenesis, so what does the virus cause blood vessels to actually grow into the cancer? Does the virus cause metastasis? So does it make the cells actually leave and go float around? And then you can go over here and say, okay, does the virus actually do that? So let's look for metastasis, so 6. And so which one has 6? So Epstein-Barr virus, which that causes glandular fever, that's in England, or mononucleosis. So if you get mono, it's caused by Epstein-Barr, which causes a ton of cancers. And I read a paper about how it was a ton of cancers are being undiagnosed. They're caused by Epstein-Barr virus, but it's too difficult to connect the two. And so the actual disease burden for Epstein-Barr virus is probably way higher as far as the amount of cancer that it causes than we're actually keeping track of, which is typical for a lot of viruses, but Epstein-Barr virus is really bad. And so it actually causes metastasis, which is really interesting. So it's clearly into the Epstein-Barr viruses, it's the LMP2A gene for Epstein-Barr virus, which codes for these proteins. Or no, I'm sorry, these are the human proteins that that gene affects, or the protein that this gene affects. Ugh. Anyway, you understand what I'm saying. These are all just different genes and the proteins and the effects that they can have, which is really interesting. See, there's Tex. See, Tex came back. Now, I want to briefly just talk about HIV. So I mentioned HIV itself. This is a really interesting paper that this article came from, or this picture. The TAT protein, which is the trans activator of transcription, it's just a protein that is made by HIV, and it has a ton of effects on the immune system, on cells. It's one of those multifactorial proteins that just does like a million things. It'll drive you absolutely crazy when you're studying HIV and trying to figure out what it does. But it can just exacerbate numerous other cancers. And so we see this really, really commonly. You can see, so here's HIV and what it does. It's suppressing the immune system, and your immune system is able to attack tumor viruses. I mean, they're viruses, right? And so with if you break that arm of your body, you know, the arm that's defending you from viruses, then it's going to be worse. And so when you have immune suppression, tumor viruses can come out and cause cancer more often. And if you want to read much more detail about that, the link is right here. One last thing, this is a really cool article that I found. It's hiding back here, so if you want to read the paper. On the left here, we have in red is like the viral stuff, and then in blue are the cancer things. And so the cancer issues caused by, this is like cellular stuff. So the S phase environment, or if you have increased genomic instability, you can have increased cancer. And so what kind of proteins from viruses are initiating genomic instability? Well, HPV E6 and E7. Oh, hey, we just talked about those. And then there's a whole bunch of other things. So if you want to sit and look at this diagram, it's really, really interesting. And then over here is really, really cool because these are the ways that your cells can defend, which is another topic that we're going to have in this class, is actual intracellular defense. And the primary article for this week is one of those, which is APOBEC. Um, and so we're going to be talking about how cells themselves, not the host immune system per se, but the actual intracellular host immune system, not intercellular, but intracellular, and how it actually defends itself against viruses using toll-like receptors and RIG. So that's like RNA interference, which we know that what we talk about, HTLV1 attacks actually affects RIG-I. Really, really interesting. And so in red, again, are the viral proteins and what they're affecting. So there's HTLV1, right, with TACs. And it's the proteins and what the intracellular defenses that they're affecting. And so down here, for instance, interferon is something that cells release when there's a virus around. And so we see, at least in the case of Kaposi sarcoma, herpes virus, we see that it's blocking interferon regulatory factors. So those are transcription factors that will go into the nucleus and induce interferon. Interferon is what cells use to tell each other that there is a virus around. And so if you're blocking that, it's going to be easier for you to infect new cells. And over here, it's really interesting. These are negative suppression from viruses, so they block MHC class 1. So that's what cells use to say, hey, oh my gosh, there's a virus inside of me. And so it makes sense that there are viruses that suppress MHC class 1. Now, it's the job of natural killer cells to kill cells that don't have MHC class 1. And so there's this constant push-pull between natural killer cells that are looking for the absence of MHC class 1 and T killer cells that are looking for virus proteins on MHC class 1. And so if you suppress this, we have a backup system is what I'm trying to say. ICAM is how cells stick to each other, and then there's a bunch of these other proteins. B-cell receptors, so that's antibodies. So Epstein-Barr virus actually increases the amount of B-cell receptors on a cell, which is to its advantage. Same with CD40, that causes activation. So that's the secondary activation signal for like a B-cell. And so if you increase that, you're going to become active, which means you're going to proliferate, which is to the advantage of the virus. Same with interleukin-6 that causes increased this is GP80 is a archaic term for interleukin-6 receptor. And so it can self-signal, and just basically what I'm trying to say is that viruses can influence their host cell to, number one, become cancerous, and then number two, to grow, and which is like the hallmark of cancer, right? It's growing. And then if they can increase that and increase those cancerous attributes, then that's to the advantage of the virus. And that's tumor viruses.